Okay, looks like we're good. All right, hello everyone. Welcome to today's installment of Paleo Perks. Today we'll be hearing from Danijela Dimitrijević from the Friedrich-Alexander-Universität of Erlangen-Nürnberg in Germany about reef communities' responses to global environmental perturbations. So a little bit about the format of today's seminar before we get started. We're starting out with some welcome and announcements. After that, we'll have the actual talk itself, and then we'll move on to a moderated question and answer session. After the question and answer session, Danijela has agreed to stick around for our tea time, which generally is just a pretty low stress opportunity to get to chat about science and non-science topics. And we encourage everyone to come to that. And then throughout the talk, this is just a reminder to please send any questions that you have for the speaker to the Paleo Perks Questions host, which is another account that is hosting this Zoom talk. So just a few kind of housekeeping things and some guidelines. So Paleo Perks values the participation of all folks interested in the paleosciences. Please remember to abide by our code of conduct during today's seminar. That code of conduct should be shared in the chat soon, so please take a look at that if you hadn't seen it. Please mute yourself during the duration of the talk. You should be muted by default, but if you're not muted, please be respectful of our speaker and keep yourself muted while the speaker is talking. Again, you can ask questions by chatting to the questions at Paleo Perks host, or alternatively, you can use the raise hand function during the question and answer time. If you have any technical issues, you should also reach out to the questions at Paleo Perks host. We do have closed captions built into Zoom. You can use the CC button to show or hide them. Additionally, we are always looking for new speakers to host in our seminar series. So you can nominate any outstanding early career researchers that you have here at this link, which will also be shared in the chat. We also have a weekly feedback form for demographic info. This is totally anonymous and optional, but it is encouraged because it helps us kind of tailor these seminars to be as inclusive as possible and to kind of serve the paleo community in the best way that we can. So you should also find that in the chat window. So finally, a bit more about today's speaker before I hand it off to her. So Danijela Dimitrijević did her bachelor's in geospatial and environmental sciences at the University of Belgrade in Serbia, then went on to do a Master of Science in Applied Ecology at the University of Poitiers in France and the University of Coimbra in Portugal, and then did another MSc in Geosciences at Friedrich-Alexander-Universität of Erlangen-Nürnberg in Germany. And she is still there as a PhD student now, studying paleobiology. Yeah, and she is going to be giving our talk today. So with that, I will hand it off to Danijela, and yeah, good luck and take it away.
Thank you, Isaiah, for the introduction. Let me just share my screen. Just a second. Can you see my screen? Okay.
Yep, perfect.
Well, perfect. Just a moment. Okay. Now I can start. Well, welcome everybody to my talk. Today I will be talking to you about the reef community responses to global environmental perturbations, which is also a topic of my PhD thesis. And let's begin by talking about coral reefs. So sclerotinia corals have been one of the main reef-building organisms throughout the Phanerozoic. They first appeared in the Middle Triassic after the Permian-Triassic mass extinction, which wiped out all the Paleozoic corals. And they established since then themselves as the dominant reef builders. And nowadays they represent hubs for the tropical marine biodiversity, and they provide a habitat for more than 1 million species. However, they are extremely sensitive to stressors such as global warming, acidification, pollution, so basically all anthropogenically induced stressors. And according to the Intergovernmental Panel on Climate Change, they are the most vulnerable ecosystems in the world. So one of the main questions I've been asking myself, is there a hope for the reefs in the future? And hopefully I can respond to that question throughout my research. But we can look at we can try to answer this question by looking at the geological record of reefs. And by observing the geological record, we can see that throughout Earth's history, reefs have gone through several episodes of waxing and waning. So we observe intervals with limited reef growth, followed by a decrease of reef growth called reef crisis, because at this time we lose significant carbonate production in the reefs. Over the past 300 million years, coral reefs have experienced 4 significant reef crises, and these reef crises coincided with hypothermal events. So here you can see the mean sea surface temperature, over time. And at the times of global warming, such as, for example, at the end Triassic and Permian, early Jurassic, or PTM, Paleocene-Miocene thermal maximum, we also notice reef crisis. And some reef crises also coincided with mass extinction events, whereas there was little extinction across other crises. And the correspondence between crisis intensity and biological turnover is little understood, particularly at the community level. So we want to understand the community changes and also their functional consequences in order to predict the long-term fate of coral reefs under climate change. To do so, we basically wanted to study the extent to which these crises induced substantial changes in reef community composition by looking at reef builders, and whether the reef crisis caused cascading effects on reef dwelling organisms. We used as reef builders, we use hypercalcifying metazoans. That's the data that we use from the paleobiology database, colonial scleractinian corals, but also extinct Paleozoic tabulate and colonial rugose corals. And then we also use the whole phylum Porifera, so sponges, because they were the dominant reef builders in the Paleozoic, and extinct Rudist bivalves that were important reef builders during the Cretaceous. And as reef dwellers, we use all reef-related organisms, minus mentioned reef builders. And we use the time frame from the Ordovician to the Pleistocene. So the global compilation of all these reef-related occurrences. And we combined our data with a rigorous novel community detection framework to identify when did novel reef communities emerge across the Paleozoic for both reef builders and reef dwellers. And why do we want to know novel community? Because these are the communities that are characterized as rapidly changed communities. They're also irreversible, so they cannot go, and they're really unique in composition and or function from all past communities. And this novel community detection framework has been first used and established in the paper by Pandolfi et al. In 2020, where they applied this novel community detection framework to marine microplankton during the Cenozoic. So the first step is to assemble compositional matrices for discrete communities, and these matrices would contain either presence of genera, and then we calculate the Bay-Curtis dissimilarity for taxonomic community composition and different for different we use it for different matrices. So first we calculate instantaneous dissimilarity, which would be faster to know than expected. So that this basically represents the magnitude of change relative to the previous state. And then we calculate cumulative dissimilarity, which is a deviation from historical baseline. So the minimum dissimilarity relative to all previous time bins. And for these two matrices, we use we do the generalized additive modeling. And when we detect outliers of the general GAMS, we identify the community as either cumulative or instantaneous. However, when they co-occur at the same time, or they match in a time series, then this community is identified as a novel community. And then all other communities in gray are the background time points. So for our data set, we use the novel detection framework for both reef builders and reef dwellers, and we identified 5 novel community events for the reef builders in the upper Ordovician, in Carboniferous, at the end Triassic, in the early Jurassic, and at the KPG. And for the reef dwellers, we identified 4 novel communities in the early Triassic, in the end Triassic, and the early Jurassic. And as you can see, the novel community for both reef builders and reef dwellers appear at the end Triassic period. So what outside of novelty events, we also wanted to study how reef dwelling and reef building community novelness correlates, and we actually see that they are tightly correlated, and this means that there is an intricate relationship between reef builders and reef dwellers, and that the cascading effects of reef crises are plausible. And this study also is important because it draws the attention to the risk of losing many more species than just corals when coral reefs vanish. But we also wanted to see what is actually happening to the reef communities after the hypothermal events. Are they able to survive this crisis? So in general, organisms can respond to stressors in different ways. One of the ways is to relocate to another area. Then second way is to adapt to stressors or to go extinct. However, more often than adapting or going extinct, we observe a species relocating to other areas. And for example, in modern organisms, we observe poleward movement of 50 kilometers per decade for marine species. And for the reefs, we basically wanted to study whether they relocate to safe areas, which we call reef refugia. So these are areas that are stable and resilient, and in the time of environmental stressors such as global warming or ocean acidification. And when it comes to reefs, the way that they can respond to global warming is they can either shift ranges to higher latitudes, or they can inhabit deeper mesophotic environment. And the big question that we were asking for this study is, are the species and the habitats they form responding in the same way? Meaning, are corals and reefs responding the same way? And luckily with Reefs Through Time, we can address this question. because we can study occurrence of reefs and corals in the mesophotic environments. And mesophotic reefs are the ones that grow in limited light conditions, and they come in two types. One type is blue mesophotic reefs. They are located in deeper waters, so normally between 30 to 150 meters. So the light is still available to photosymbiotic corals there. And then another type are brown mesophotic or turbid reefs. And as you can see here on the picture, they really do look kind of brownish. And when it comes to brown mesophotic reefs, we can easily detect them in geological record by assigning coral or reef occurrences to siliciclastic environments. But when it comes to deeper water, it's really hard to or impossible to establish the absolute depth of reefs and corals. That's why we use bathymetry estimate relative to the wave base, as it is also reported in the Paleo Reefs database. So we can have three categories: ones that are above the fair weather wave base, below the fair weather wave base, and below storm weather wave base. And essentially everything that is located above the fair weather wave base would present euphotic or shallow water, while everything that is below fair weather wave base is deeper water. And when it comes to the observation from the fossil record, it is very interesting that the first post-crisis reef at the end Triassic was a coral microbial reef that grew below the wave base, and it was growing in mesophotic environment and in subtropical setting. So we wanted to see how prevalent this or is there a pattern for this kind of establishment of reefs post-crisis and post-hyperthermal events. And we hypothesized that in the aftermath of hyperthermal events, reefs and corals sought refugia in mesophotic settings and or in higher latitudes. And for this, we use two different data sets. So for reefs, we use the Paleo Reefs database, and for corals, we use the occurrences from the paleobiology database. And here you can see a number of reef sites through time for the past 300 million years. And here you can see sampled in being colonial coral genera for the past 300 million years. So we use colonial coral genera because they are reef builders. And in orange, you can see the marked important hyperthermal events that coincide with reef crisis. So that would be the end Permian, Triassic, Jurassic, Toarcian, ocean anoxic event, and Paleocene-Eocene thermal maximum. And it is important to note that there was an absence of reefs and also of scleractinian coral genera in the earliest Triassic. So first, we looked at the proportion of coral reefs in the euphotic and blue mesophotic zone. And here again, we can see this early Triassic gap when there is no data. But we observed that after the hyperthermal events, there is an increase in the proportion of reef sites in the deeper mesophotic environments. So we wanted to check this quantitatively as well, and we observe. We did the analysis based on whether the reefs are observed in normal times or in post-hyperthermal times. And when I say post-hyperthermal, I mean the stage after the hyperthermal event. And then we compared the reef proportion in the euphotic shallow water and in deeper blue mesophotic water. And what we see, and it's really striking, is that in the blue mesophotic water, the reef proportion increases in the post-hyperthermal times. When it comes to brown mesophotic, we don't see much difference between the normal and post-hyperthermal times. So we can say that reefs sought refugia in the deep. So we see an increase of mesophotic reefs in the aftermath of hyperthermal events, but they didn't build reefs preferentially in turbid water, or they didn't seek refugia in the turbid water. When it comes to range shifting, we also did analysis at normal time and post-hyperthermal time for reef sites. And this is showing the absolute paleolatitude. And we observe in the post-hyperthermal times 8.2 degrees poleward shift. And this means that the higher latitude served as refugia in the aftermath of hyperthermal events. But what about corals? The question that I mentioned before, are species responding in the same way as habitats they form? Interestingly, the corals do not seek refugia, or they didn't change much between the normal and post-hyperthermal times in blue mesophotic water. However, the proportion of corals did increase in the turbid or brown mesophotic water in the aftermath of hyperthermal events. When it comes to latitudinal range shift, we observe similar pattern as for reefs. So even 9 degrees latitude poleward shift for coral occurrences in post-hyperthermals. So they also sought refugia in the mid-latitudes. So in order to understand more depth the ecological effects of crises in the past, we also studied the evolution of photosymbiosis and the photosymbiotic efficacy of corals. Corals in tropical reefs and their ability to build massive complex structure is actually the consequence of their symbiotic relationship with algae called zooxanthellae, which are embedded in their tissue. And these algae contribute up to 90% of corals' nutrition and energy in exchange for protection and inorganic nutrients. Another group of corals are azooxanthellate corals that do not have symbionts, and they rely almost completely on a heterotrophic diet, and they are typically solitary. And they can also form colonies. And the evidence for photosymbiosis in corals is difficult to infer directly because algal symbionts are not preserved along with fossil coral skeletons. And over the past decades, a number of different methods have been developed to solve the so-called photosymbiotic mystery. But among these morphological traits are, I would argue, the best way to study photosymbiosis in corals, because they're easily preserved in the fossil record and they're also regularly reported in the literature. And they also reflect the photosymbiotic efficacy because zooxanthellate corals require the most efficient light harvesting strategy, so their morphology would exhibit the photosynthetic adaptation. And some of the morphological traits that have been used to infer the photosymbiosis are a corallite diameter. So corallite is a skeletal cup in which the polyp sits. And then we can look at the way the complexity or the arrangement of corallites in a colony, so-called corallite integration. Also, we can look at the growth forms or degree of branching, because, for example, almost all zooxanthellate corals grow large and form massive structures. But for this study, we focused on the corallite diameter as a proxy for photosymbiotic efficacy. It is a quantitative objective approach, and corallite sizes are used as a proxy in a way that smaller corallite sizes are associated with higher photosymbiotic efficacy, while larger corallites are assumed to be less dependent on photosymbionts and to maybe rely more on heterotrophic feeding. And in this study, we used two databases, global compilation of fossil coral traits that we developed here in Erlangen, called ART database, Ancient Reef Traits database, that is made to mirror the database called Coral Traits database of modern modern coral traits. And we applied various analytical methods to understand how photosymbiosis has evolved based on the corallite sizes. But here I will highlight the one that help us understand the ecological patterns, and this is based on the abundance or data or the occurrences of its occurrences weighted data of coral genera. And we can see that over the past 250 million years, there was a significant decreasing trend in mean corallite width of colonial corals. And this suggests that corals with smaller corallites became more dominant over time, thereby obtaining a competitive advantage in shallow tropical nutrient-poor seas they largely occupy today. So they basically improve their photosymbiotic efficiency. So to summarize my talk, we observed novel communities that they emerged after reef crises and after mass extinctions, and intricate relationships between reef builders and reef dwellers rendered the cascading effects of reef crisis plausible. We also observe renewed reef building in the mesophotic zone and mid-latitudes in the aftermath of hypothermal events and reef crisis. And we also observed that colonial corals became more reliant on photosymbiosis over time, obtaining competitive advantage. However, it would be very nice and useful to extend this study to trait-based approaches, and I believe they are a way forward to understand the ecological effects of crises in the past, but also that they will help us project responses of reef-building coral. Thank you very much for your attention.
Awesome. Thank you so much for that awesome talk, Danijela. That was really, really, really great to hear. So yeah, just a reminder, if anyone in the audience has questions, you can send them to the PaleoPerks questions host. But we already have a few audience questions that came in. So if it's all right with you, Danijela, we can just jump right into it.
Sure.
Okay. So I will both read the question and also paste it in the chat box so everyone can read it. Okay, so our first question: how applicable is the fossil coral record and past climate events to modern corals and the modern climate? And I guess that extends to kind of modern climate change. So maybe that's a big question to start us off, but
it's a very big question. So there is a study by Nusseibeh Raja et al. From 2021, I think, where they use machine learning to study the extinction risk of modern corals based on the extinction of the fossil corals. So I would say it's applicable. So we just need to use very sophisticated methods such as machine learning.
Yeah, definitely. Thank you. Okay, another question. Okay, this question is from Elizabeth Dowding. Reefs keep evolving. Why do you think these community structures are so successful?
I think today they might be successful because of the photosymbiosis, but in the past I'm not so sure why they were so successful. Maybe because of the co-occurrence with other organisms, but this is I'm mostly focused on why they were not successful than to answering the question why they were successful.
Yeah, definitely. Okay, we have more questions coming in. Okay, so the next question. Does corallite diameter vary within a single species given different ecological conditions, or is this proxy only effective when comparing different species?
No, there are some studies that correlate diameter. It's not just dependent on the photosymbiotic efficacy, but it can also exhibit the corals can exhibit this phenotypic plasticity where they adapt to the environment. So, for example, if you have a high sediment input, you will have smaller corallites, or for example, if the corals are growing in deeper water, they might have bigger corallites. So sometimes it's also variable within species as well.
Okay, very interesting, cool. Okay, we have another anonymous submission here. I'll just keep firing them off. Could benthic foraminifera be used as a depth indicator for your fossil coral bathymetries? Are they found alongside your corals? Could the changes in carbon isotopes, delta C13, of the coral calcite be used as a proxy for photosymbiont occurrence akin to planktic foraminifera?
Yeah, definitely. I did not use forums as a depth indicator, but this is a very interesting idea and maybe something that would be useful to use in the future.
Yeah, definitely. That seems like it would be a cool way to kind of, add just like one more kind of piece of data
Yeah.
to the puzzle.
Yeah, exactly. I don't think well, I'm still in my PhD, but in the future definitely we should be studying more organisms, like the whole communities, because everything is related, and obviously we can use one organism as a proxy for another.
Yeah. Okay, next we have a short one. This is submitted by Roy Plotnick. Would Paleozoic corals have been photosymbiotic?
There are studies that show that tabulate corals might have been photosymbiotic, but to be honest, this is not my area of expertise. I've read the papers, but I don't know if they were exactly. But as they grow massive and colonial, if we use the morphological traits, then yeah, we can assume that they were.
Interesting.
I don't know about the rugose corals.
Yeah. Cool. All right. We've got more questions coming in, if that's okay with you, Danijela. I think we have an interested audience today. So maybe this kind of falls under the category of like a bigger question. What does the emergence of novel communities after reef crises and mass extinctions mean in our modern time of climate change? And so I guess to kind of extend on what they're asking, how can we kind of, extrapolate what we see in the past to the present?
I think just observing the community change is not enough, and this is something that I want to do in the future during my postdoc, is to look at the functional changes or functional emergence of novelty, because I think this is more relevant than to observe just community change. Because yeah, if we observe a completely novel community, it does not tell us anything for the future, but we want to see how this community's function changes, and whether the function of the reef itself will be preserved for the future.
Interesting. Cool. All right, we've got another question here. This one is anonymous. Concerning novelty, how long does a novel reef community take to establish after an extinction event? How does preservation relate to reef identification in deep time?
This is really hard question. How long? I don't know how long, because we identify novel communities. So if there is, as I mentioned before, missing, if there is a reef gap and coral gap at the end Permian, after the end Permian, at the beginning of the Triassic, we actually observed the novelty from the last Permian stage to the first occurrence. So we basically skip the stages that don't have any data. So it all depends on how long the stages are and how much data there is.
Mm.
How does preservation relate to reef identification in deep time? I'm sorry, I don't know how to answer this question.
Yeah, it's a thinker for sure. I guess it's like, yeah, it really just depends on, like, how the, you're kind of like a lot of things, how you're kind of doing the analysis and how you are collecting data and that sort of thing. And
I mean, preservational biases are ever present everywhere, and they need to be addressed, that's for sure.
Yeah, but that was great. Thank you for the answer.
Thank you.
Okay, got a few more questions still rolling in here, if that's okay with you. So this one was also submitted just anonymously. Great talk. How long, months or years, did the relocation of coral reefs take post-hydrothermal events? Do you anticipate a similar length of time for reef relocation with respect to anthropogenic climate change?
How long? Months or years? Well, I can't say that, because we also study the stage level, so it's really hard to tell months or years in the fossil record. But given that we have today on a decadal scale on modern organisms 50 kilometers poleward shift of marine species on average, yeah, I would say it doesn't take much long for at least for the coral propagules to migrate, but coral reefs they can't migrate, obviously. It takes a very long time for them to establish and to grow.
Awesome. I think we are coming to the kind of end of our kind of submitted questions. I think we have one left. So in case anyone else has any last minute questions they want to submit, I guess we have one more for you, Danijela, if that's all right.
Okay.
Okay, so here we go, posting it in the chat box now. Why did corals in the more turbid mesophotic zone respond differently to the less turbid mesophotic zone? Or do you have any ideas or
why did corals in the more turbid mesophotic zone respond differently to the less turbid mesophotic zone? I'm not sure I quite understand the question. So the corals in the post-hyperthermal events, they did increase So the occurrences of corals did increase in the turbid zone, and it might have been because in the turbid zone, they are more protected against bleaching because of the suspended sediment particles. But I don't know why did they respond differently to the less turbid mesophotic zone. Well, that might be the answer. Maybe they are protected against bleaching there. But it's very interesting that reefs do not follow the same way as corals in the post-hypothermal events. So the corals do seek refugia in the turbid zone, but they're not able to build reefs there. So, yeah.
Very interesting. It's a really interesting, I mean, corals, of course, are interesting, and then reefs more broadly are kind of like interesting systems. And there's still so much that we as a scientific community have yet to figure out, So
yeah, definitely.
It's really exciting research. So
thank you.
Well, yeah, thank you again for your talk. I will just kind of wrap us up with our kind of closeout slide. So thank you everyone for joining. Yeah, if you haven't done so already, we would appreciate it if you could fill out the weekly feedback form with a link that has been shared in the chat, just so we can learn a bit more about who's in attendance at today's seminar. Yeah, and we always appreciate that. Just a little plug for our next seminar. It's called Mechanisms for Ocean Carbon Storage and Glacial Climate Model Simulations with Malin Ödelin from the Geomar Helmholtz Center for Ocean Research in Germany. So we hope to see you all then. So with that, up next, Danijela has graciously agreed to join us for a casual kind of tea time. So we encourage that anyone who wants to stick around and just kind of say hello or chat or ask kind of questions in maybe a more personal setting, whether they're related to science or not. Yeah, that's a great opportunity to do that. And we always have a question of the week that tries to match the theme of the talk. So this week's question of the week, not just for Danijela, but for anyone who's at tea time, is what is your favorite reef building organism, past or present? So I'm looking forward to some exciting discussion there. So yeah, with that, we'll have a little 2-minute break so you can get up, grab some water, stretch your legs. Of course, if you have paleo pets, they're welcome to join, as always. And we'll see you then. So yeah, see you all in 2 minutes. And I started the timer. So